Hey everybody, and welcome back to the HIV RNA Test Guide podcast. You know us. You're a trusted source for accurate HIV testing all across the U.S. And today, we're doing a deep dive into some pretty cutting-edge HIV vaccine research. Specifically, a recent discovery that honestly kind of blew my mind.
Oh, yeah.
So, to set the stage a little bit, when we talk about vaccines, all right, so get this. We're looking at some pretty wild HIV research today. This paper from Science Immunology has got everyone talking, and
Yeah, it's really interesting.
it's kind of mind-blowing. Basically, imagine your immune system, right, all geared up to fight HIV, but then it accidentally starts attacking itself.
It's like, whoa, that's not what we're going for.
Yeah, wild, right.
Yeah, it really highlights how complex the immune system is. The researchers at Scripps, they were looking at antibodies produced after multiple doses of an experimental HIV vaccine, and what they found was completely unexpected.
Really? So what happened?
Instead of directly attacking the virus, some of the antibodies were, get this, latching onto other antibodies that were already bound to the HIV.
There's like a chain reaction of antibody.
Yeah, antibodies against antibodies.
Okay, hold on. Let's back up for a second.
Yeah.
We know most vaccines work by introducing like a harmless piece of a virus, right? And that way, our bodies can kind of learn to recognize it and fight it off. But why is HIV such a tough nut to crack? And how does this new discovery change things?
Well, you see, HIV is really good at mutating. It's like it's constantly changing its disguise, which makes it super hard for the immune system to keep up.
It's like it's always one step ahead.
Exactly. And that's why developing a really effective HIV vaccine has been such a challenge for so long.
So this research is like, it's adding a whole new layer of complexity with these antibodies against antibodies.
Right, exactly.
So the big question here is, does this weird antibody behavior, does it help or hurt the fight against HIV? Like, is this a good thing or a bad thing?
That's the million-dollar question, isn't it? Researchers are still trying to figure that out.
So what are they thinking?
Well, one possibility is that these extra antibodies could actually be a good thing. They might create larger immune complexes, and those complexes could actually trigger a stronger immune response overall.
So it's like sending a bigger signal flare to alert the immune system.
Yeah, like, hey, look over here, there's a problem.
Right. But then again, couldn't this also backfire? Couldn't these extra antibodies be kind of distracting the immune system from actually fighting the virus directly?
Yeah, that's the other side of the coin, for sure. Instead of this, focused attack on the virus, the immune system might get bogged down in this whole antibody chain reaction, and that could end up leading to a less effective response overall. It's a tricky balance.
Like you said, it's super complex.
And scientists are really trying to sort out these potential benefits and drawbacks, to see which way it tips.
Yeah, because it could go either way.
Exactly.
So it seems like understanding exactly how these immune complexes, how they form and how they function, that seems to be the key here. Can you kind of break down what they are and how they actually play into the immune response?
Sure. So basically, an immune complex it's essentially a clump of antibodies bound to their target, which in this case is another antibody that's already attached to the HIV virus.
Okay, so it's like a cluster.
Yeah, it's a cluster. And the size and the composition of these complexes, they can really influence how the immune system recognizes and responds to them. Larger complexes might be more easily detected and gobbled up by immune cells.
So that would lead to a stronger immune response.
Potentially.
So the size of these immune complexes, that could be like a make or break factor in whether this antibody chain reaction is helping or hurting.
Exactly. And that's why researchers are using all these fancy imaging techniques now to actually visualize these complexes at a really, really tiny level.
Wow, so they can actually see these things.
They're trying to, they want to understand how the shape, the size, the composition of these complexes, how all of that affects their interaction with different immune cells.
Wow, that's incredible.
Yeah.
This research, it has some pretty major implications for how we think about HIV vaccine design, right? Like this could really change things.
Absolutely. I mean, this discovery is definitely making scientists rethink their strategies. Instead of just focusing on triggering those antibodies against HIV, now they're looking at ways to either encourage or suppress this secondary antibody response.
So it's like they're trying to fine-tune the immune response, but almost like adjusting the settings on a machine to get the perfect output.
Yeah, that's a great analogy.
Yeah.
And part of this fine-tuning involves experimenting with different vaccine schedules, changing the timing, the types of doses given to see if it influences this whole chain reaction.
So it's like finding the perfect recipe.
Right, exactly. It's got to be precise.
This is all so fascinating. This research is a great example of how even unexpected discoveries can open up these whole new avenues of exploration, right? It really shows how important curiosity-driven research is.
It really does, yeah. Sometimes the most groundbreaking findings, they come from asking those unexpected questions and being open to those surprising results. It's all about pushing the boundaries of our understanding and challenging what we think we know.
And this whole discovery, it feels like a major plot twist in the whole HIV vaccine story. I'm really eager to see where this goes next.
Me too. It's really exciting.
But before we dive into that, the potential impact on treatment and prevention strategies, let's zoom out for a second and look at the bigger picture.
Okay, yeah, let's do that.
What do you think?
I agree. This research, it raises some really fundamental questions about how our immune system learns and adapts. Could this antibody on antibody action be more common than we initially thought? And could it even be happening in other diseases?
That's wild. I mean, if that's true.
It could be a whole new way of thinking about the immune system.
Exactly. And I can't wait to dig into all of that with you.
Let's do it.
But first, let's take a quick break, and then we'll be right back to explore the potential impact of this discovery for HIV treatment and prevention.
So before the break, we were talking about how these antibodies that target other antibodies, they might actually be a key player in the immune system's response to HIV. And the research, it's mainly focused on vaccine development, but what about treatment? Could these antibodies be used to actually fight HIV directly in people who are already infected?
That's a really good question. I mean, it seems like a logical next step, but I'm guessing there are some challenges there.
Oh, for sure. While the research is initially focusing on vaccines, scientists are definitely exploring the possibility of using these antibodies for treatment, Imagine if we could somehow reprogram these antibodies to recognize and attack HIV-infected cells directly. That would be huge.
It would be a total game changer for HIV treatment.
Yes, exactly.
But is that even possible? Can we actually, manipulate antibodies in such a specific way?
With the advancements in biotechnology and genetic engineering, it's becoming more and more possible to modify antibodies for therapeutic use.
So it's actually doable.
We've seen amazing progress with antibody-based therapies for other diseases, like cancer, for example.
So it's not just science fiction anymore.
Exactly. The challenge with HIV, though, is really understanding the specific mechanisms of these antibodies and how they actually interact with this particular virus.
So it's kind of like trying to hack into a really complex computer system, right? You need to understand the code before you can make any changes.
Yeah, that's a great analogy, and that's why this research is so crucial. The more we learn about these antibodies and their interactions with HIV, the better we'll be able to harness their potential for treatment.
This really highlights how important basic science is. These seemingly obscure discoveries can have such big implications down the road.
Absolutely. Basic research is the foundation for everything. It's about exploring the unknown, asking those fundamental questions, and sometimes, stumbling upon answers that can revolutionize entire fields.
And this whole discovery about antibodies targeting other antibodies, that seems like one of those paradigm-shifting moments, for sure. Speaking of shifting perspectives, let's talk about HIV prevention. Do you think this discovery could lead to new strategies to prevent transmission in the first place?
Oh, absolutely. I mean, we already have PAP, which is very effective in preventing HIV transmission, but imagine if we could develop a new kind of PAP that uses these antibodies to neutralize the virus even more effectively.
So current PAP medications, they work by blocking HIV from entering cells. Could this new approach involve, creating antibodies that directly target the virus, almost like a preemptive strike force?
Exactly. That's the hope. If scientists can successfully manipulate these antibodies against antibodies, we could potentially have a PAP strategy that provides even greater protection against HIV infection.
That would be a huge breakthrough, but we're still in the early stages, right?
Oh yeah, definitely early stages. We need a lot more research to see if this is actually feasible.
Makes sense. We have to be cautious and do things right.
Absolutely. We need rigorous research to ensure it's both safe and effective.
It's incredible to think about how much we still don't know about HIV and the immune system.
I know, right? The human body is incredibly complex, and HIV, with all its mutations and tricks, it's a tough opponent.
It's like a constant arms race.
But with each new discovery, we get a little closer to understanding how to fight it.
And this research also shows the importance of collaboration in communication and science.
Mm.
You know, sharing findings, working together, challenging each other's ideas. That's how we make progress.
Absolutely. Science thrives on collaboration. It's all about sharing knowledge, building on each other's work, and constantly questioning everything. And with HIV research, global collaboration is absolutely crucial.
Now, before we move on to the ethical considerations surrounding this research, I want to go back to something you mentioned earlier. You talked about the possibility that these antibodies might be used to treat other diseases besides HIV. Could you expand on that a bit?
Sure. So while the focus now is on HIV, the principles behind these antibodies and their ability to target other antibodies, that could potentially be applied to other infectious diseases or even autoimmune disorders.
So this discovery could have much broader implications beyond just HIV.
Exactly. It could be huge. It really shows how scientific breakthroughs in one area can often have ripple effects across many different fields of medicine.
It's like opening a door to a whole new world of possibilities.
It's a reminder that scientific exploration is full of twists and turns. We might start with one goal in mind, but sometimes the most important discoveries happen in unexpected ways.
And that's what makes it so exciting.
Absolutely. It's all about embracing the unknown, being open to new ideas, and pushing the boundaries of what we think is possible.
Welcome back. So we've been talking about this research on antibodies that target other antibodies in the context of HIV. We've talked about the potential benefits, and drawbacks, and even touched on potential applications for treatment and prevention. But now let's kind of shift gears a bit and talk about the ethics of all of this. I mean, it's one thing to explore the science, but we also need to consider the implications and do things responsibly, right?
Oh, absolutely. Whenever you're talking about messing with the immune system, especially in such a complex way, you got to think about the ethics, proceed with caution, weigh the potential risks and benefits.
For sure. So what are some of the main ethical concerns that scientists and ethicists are raising about using these antibodies?
Well, one of the biggest concerns is the potential for unintended consequences. Like we've been saying, the immune system is incredibly complex, and we're still learning how these antibody interactions really work. And if we start manipulating these interactions, whether it's for treatment or prevention, there could be some unexpected effects, both good and bad.
So it's kind of like we're entering this uncharted territory, and we need to be ready for anything.
Exactly. So one possibility is that manipulating these antibodies could trigger an immune response that gets out of control, leading to harmful inflammation or even autoimmune reactions.
That'd be bad.
Yeah, and then there are the long-term effects to think about. Something that seems beneficial in the short term could end up having negative consequences down the line.
So it's like we got to be really careful.
We need thorough testing, preclinical testing, and carefully designed clinical trials to make sure we minimize those risks.
Absolutely. So rigorous testing in controlled settings is key before we even think about human trials. And even then, transparency and ongoing monitoring are crucial to make sure the participants are safe.
Right, exactly. We have to prioritize safety every step of the way.
Another ethical consideration is access. If treatments or preventive measures come out of this research, how do we ensure everyone who needs them can actually get them, regardless of their income or where they live?
That's such an important point. Scientific breakthroughs don't mean much if they don't reach the people who need them most. We have to work on breaking down those systemic barriers to healthcare access, make sure that cost, location, those kinds of things don't prevent people from getting potentially life-changing treatments.
It's a tough issue for sure, but it's a conversation we need to have alongside the scientific progress.
Absolutely. Access and equity have to be part of the conversation from the beginning.
Okay, so let's zoom out a bit more and look toward the future. What are the next steps for scientists who are studying this antibody-on-antibody phenomenon?
Well, I think one of the main priorities is to really understand how these antibodies work on a deeper level. Researchers are using all kinds of advanced imaging techniques and computational modeling to get a detailed picture of these interactions at the molecular level, like creating a roadmap of the immune system.
So it's like they're trying to crack the code, understand the language of the immune system.
Yeah, exactly. And once we have a better understanding of this language, we can start exploring ways to manipulate it for therapeutic or preventative purposes.
And that'll involve like lab experiments, animal studies, and if things go well, eventually human clinical trials.
Exactly. It's a step-by-step process, and it requires a lot of rigor, transparency, and collaboration among scientists from different fields. We're seeing immunologists, virologists, bioengineers, computational biologists all working together to solve this puzzle.
It's really amazing to see all these different experts coming together.
It really highlights the power of interdisciplinary collaboration.
Okay, so before we wrap things up, I want to leave our listeners with something to think about. We've been focusing on the potential of these antibodies to fight HIV, but what if this discovery tells us something even bigger about the immune system itself? What if this antibody-on-antibody action isn't just specific to HIV, but is a more general mechanism that our immune system uses to regulate itself, like fine-tuning its response to different threats?
That's a fascinating question. And, researchers are actually exploring that possibility. It could be that this phenomenon, first observed with HIV, is actually much more widespread and plays a role in other infectious diseases or even autoimmune disorders or even cancer.
Wow. It's like we've stumbled upon this hidden doorway in the immune system, and now we're just beginning to see what's on the other side.
Exactly. And this discovery has opened up a whole new chapter in immunology. It's really exciting to think about where it might lead. It's a good reminder that scientific exploration is full of surprises. Even when we think we have things figured out, nature always has a way of showing us something new.
Well said, and that's what makes science so cool. So we've reached the end of our deep dive into the world of antibodies against antibodies. We've covered the initial discovery, talked about the potential implications for HIV treatment and prevention, discussed the ethical considerations, and even looked at the broader possibilities that this research opens up.
It's been a great discussion, and I hope our listeners have gained a better understanding of how complex the immune system is and how much we're still learning, not just about HIV, but about human health in general.
Absolutely. This research is a testament to the power of curiosity and collaboration, and it reminds us that breakthroughs can happen in the most unexpected places. And that's why we need to keep exploring, keep asking questions, keep pushing the boundaries of what we know. So thanks for joining us on the deep dive, and until next time, stay curious.